another minute or two to call in since it's just 1 minute till. Oh, now it's 3 o'clock, but we'll give people a little bit of time here. All right, so I have a call-in user 2 listed in participants. Who's here?
Hi, Dr. Hinkie. This is Zelaya. Can you hear me?
Yep, I can. Hi, Zelaya.
Hey, how are you?
Good. How are you?
I'm good, thanks.
All right, we're going to go through the lab objectives, and I'm just going to go right down the list. So, you don't have to, since it's only one person so far. As long as there's no background noise, you don't have to mute. Just jump in and stop me when you have any questions. All right?
Okay.
All right, let me share these objectives.
And I wanted to video. I don't know if you have a presentation up, but for some reason it wouldn't work for me.
Oh, okay. Did you use the call-in number or the? So there's a call-in number on there, and there was also a URL.
Yeah, I use the URL, the meeting link, and then it gives me like this error. Yeah, I used that first, and being that didn't work, I just decided to call in.
Okay. Oh, sorry about that. I'm not sure why that link didn't work. That should have worked right into my classroom or into my WebEx room.
Okay. But where can I find those? That should be in the under, let's see, what you're looking at, the lab objectives.
So the lab objectives are in top hat at the start of each lab. They list them for each lab. So, and what I just did was I just cut them all out into one document. But what I can do is post those in our classroom, so you'll have the checklist.
Okay, sounds good. I'm trying to multitask here and head on over the top hat, but okay, thank you.
Yep. So if you just go right into our classrooms, are you at your computer now?
I am, yes, ma'am.
All right. So what I'll do is I will just put those. It's moving a little slow, but I will put those, if you click under Lab Part 1, Lab Exercises, I will put those in there in just 1 second. Let me save these here. Lab Objectives Practical 1. All right, if you go into the Lab Upload Create, upload a file. If you go into the Lab Part 1, upload these right into here now. Lab Objectives Practical 1. All right. And sure, I picked Lab Objectives Practical 1. I picked the wrong. Those are old. Let me delete that file because those are last year's. I should have looked at the date on that. they are similar, but there's something since we aren't in the lab that you're not doing. So save as.
Oh, okay. I see where you those are the one you're about to remove. Lab objectives, practical one.
Yeah.
Okay.
Let's see. There we go. And of course, my battery on my mouse just died.
Mm.
Right, let's see. We're going to get rid of those and put the right ones up here.
Okay.
File. Aren't they where I saved them? All right. Okay, so those should be in there now. They load at the very bottom of the page. So if you just scroll down the page, you'll see
Okay.
that document.
Okay, yeah. 2020. Got it here. Okay.
All right. So
Cool.
those are the ones that those are in the right order. That's for our class.
Okay, thank you.
You're welcome. That's when I saw on here I was going to load them, and I saw that it said, oh, those are already they come at the top of each top hat.
Mm-hmm.
But yeah, that's easier if everybody has those right there instead of going in. So what we'll do is I'll just go right down the list,
Mm-hmm.
and then yell if you have any questions. So the first one for the lab safety objectives, we're not in a lab, so so some of these don't apply because we don't have a location for a fire extinguisher and eye wash station. We're doing virtual labs.
Mm-hmm.
Just managing time effectively, knowing, the names of the equipment, things like that. So let's skip right through those and go to the scientific method. And the first objective, a lot of these are knowing and using the vocabulary appropriately. So know the steps of the scientific method. That it always starts with an observation. And then know those definitions, hypothesis and inference, using qualitative versus quantitative data. So if we can count it or measure it, it's quantitative. If we can put a number to it. If it's some quality that we can describe, like a color, then it's qualitative.
Mm-hmm.
The variables. So this is probably one that people struggle with the most.
Mm-hmm.
So looking at that experiment, my best recommendation for how to be certain on those is to try to write an if-then statement for your hypothesis. So your hypothesis, when we say, oh, it's your best guess of the outcome, this is what you're predicting is going to happen. So you can state that prediction as, if I do this, then here's what's going to happen. So by doing that, it tells you which variable's which. So what you're doing when I say, if I, keep all the I's together. If I, there's two I's, that's the independent variable. If I eat cookies for breakfast, then my grades will improve. So my grades depend on what I have for breakfast. So that's the dependent variable, is what comes after the then. So if you can phrase it like that, it makes it a little bit easier. Sometimes it can be challenging to figure out exactly what you are controlling and changing, right? But the thing that changes in response to what you change is going to be is what depends. What your outcomes are, that depends on your independent variable. So what you're recording as data is your dependent variable. So that's how I try to figure them out. Is and I mean, it's something that coming up with a hypothesis that tells you exactly what your experiment is. That's what we strive for, and it's not as easy as it sounds. That's why you get to learn about the scientific method in every science class ever, because sometime around grad school you say, "Oh, now I get it." So so if it's not clear, if you struggle with it, that everybody does to begin with, it's clear. But that's kind of the easiest way, I think, is making it an if-then statement, because it tells you, if I do this, that's my independent variable, then, sorry, my dog's barking in the background, then something else will happen. That's my dependent variable. And that tells you what you have to do. If I eat cookies, then my grades will improve. Well, that tells me my experiment. I have to eat cookies and then take a test to see what my grades are. So it designs your experiment for you when you do it that way. And then the other things that students tend to confuse is because they have them, similarly named as controlled variables versus control groups. So controlled variables, just focus on that, it says variable. So those are things that could change, and they could change the outcome. So in my cookie example, if I think of anything that could change the outcome, what are all the things that could affect your test grades? Sleep, what textbook I use, what instructor I have, how much time I study, how much sleep I get. All of those things could affect the outcome, my test grade. So to make sure that I'm only looking at the impact of those cookies on my test grade, those variables, all those things that could change the outcome, all have to be held constant. So they are controlled variables. So they are things that could change, but I'm not going to let them. And then the control group is, this is who I'm going to compare my results with. So my control group, I have an experimental group that I'm going to change the independent variable for. They're going to get cookies. And then my control group, I'm not going to change that independent variable. They're not getting cookies. So those are the two differences, so that I can compare and see, well, what would have happened anyway? So maybe my test grades would have gone up anyway, even if I hadn't eaten cookies. So I have to have something to compare to, and that's my control group. And then I think it was when we did the macromolecules, we really had a positive and a negative control, so you could look at that. Not all experiments have a positive and a negative control, but they all have a control group. A positive and negative control are when you know what a positive result looks like, and you know how to get a positive result, and you know what a negative result looks like, and you know how to get a negative result. So I know if I have my Biarritz reagent, and I know it turns purple if there is protein present in a solution, then I can have a positive control by just having a solution with protein that I know I'm in charge. I made that, and I know it has protein in it, so I know I'm going to get a positive result. So that's my control, my positive control. I add the Biarritz to my protein, and sure enough, it turns purple. So it validates my experiment. It says, yep, the reagent works just the way it should. And whenever I see this color, it means a positive result. And then similarly, I know that Biarritz only responds to protein. So if I put it in water, I'm not going to get a color change. So I can take some water, put Biarritz in, and sure enough, I don't get a color change. This is my negative control, and any of my test results with my unknown subjects that look like that are negative. So if I know what each of my variables is supposed to do, how things react, I can select and design a positive and negative control. I can't always do that if I don't know the outcome. So like I wouldn't know what the outcome, what the effect of cookies on a test grade is. So there's no way to design a positive and negative control for that. That's just a control. Does that make sense?
Yes, it does.
Okay. And then that analyze and interpret data from graphs and tables. I think everybody's doing pretty good on that. You know, the x-axis, the horizontal axis, is where I'm going to put my independent variable, and the y-axis is going to be where I put my dependent, and then that should show me trends so that I can, even if I measure, like say I'm measuring pH and I measure it at 3, 5, 7, 9. So I have 4 points I measured it at, but if I connect those dots in a line graph, it lets me predict what would happen at 4.5, what would happen at a pH of 5. So I think that one is pretty good. Let's see. Any questions on scientific method?
No.
All right. Measurements. So measurements, look, when we're in the lab, we use instruments that are already in metric. So it's just reading and recording. So a lot of the activities we do are just so your brain has sort of some relative understanding. Like I know if somebody says 1 meter, that's just about a yard. So I can kind of do this quick conversion in my brain or have some idea of how big that is. But then when we get to the smaller scales, it's just easier to work in metric and measure in metric and do all my conversions in metric. And all it is moving the decimal place. So if I'm converting among these groups, so I start with that base is in the middle, whatever my base is. If it's a liter, then I can go smaller, like decimeter. There are 10 of those in 1 liter. Centiliters, there are 100 in 1 liter. Milli, there are 1,000. So all I have to do is move my decimal 1 for each of those. So remembering what the prefixes mean is important. We have all those, but really what we're going to use mostly is kilo, and then centi and milli for the smaller ones. That's what we use primarily in the lab. And the kilometers, I guess for grams we could use that in the lab, but just remembering like a 10K race, that 10 is 10 kilometers. So that's units. And then going the other way are smaller. So that tells you which way.
Oh, hey, it's breaking up just a little bit. Can you still hear me? Oh, I can't. Wow, I can't hear. Oh.
Uh-oh. This isn't going well. There we go. All right. I think we're still recording. And I think I lost you there. I lost connection for a little bit, but now it seems to be back. So I'll just keep recording this. And let's see. We have, oh, nope. All right, we've lost everybody from the meeting. I will keep recording this and post it. So, sorry. Something seized up on my end, and that was it. So, Elia lost the audio. Well, I cut out her audio when my computer seized up here. So we will continue, and I'll just post this recording. Sorry, everyone, for all the technical difficulties. Everyone should know that the meniscus is the low point when I have that water in my graduated cylinder that creeps up the side because of adhesion. The low point, that's where I'm going to read my measurement in my graduated cylinder. Should know the different units. So the instrument you would use to measure length, meters, mass, grams, volume, liters, and temperature, Celsius. And there, you got back in?
Hi, Dr. Hinkie. I made it back in. It was terrible. But I couldn't make out anything you were saying. I left off at the kilo, but that's okay. I'm following.
Okay, well, good. I know. You know what? My computer's up on this end, and everything went haywire over here. I couldn't do anything. So that's I kind of just readjusted, and I didn't know if I kicked you out or if you hung up to try to come back in. So
Yeah,
I'm glad you made it back in.
Thing, but I think I pretty much I don't necessarily have any questions on it. But I have the objectives, and I'll pretty much go over through the top hat. I do wish, I guess there is a way, because when you grade them, is there an answer key at all on the top hat? Like we got wrong.
I don't know if the answers show up on yours. So they don't actually show up anything wrong. So we've had issues in the past of people sharing answers with other classes or sections or passing it on. So we don't show any of the answers. So what we do is that people who do want to know what questions specifically they got wrong, you can just give me a shout, and I can go over those, go over the areas that you want to look at a little bit more. So we can do that on any of those. So for any of the students who want to do that, want to know more specifically. And I've been trying to just at least show in the quizzes, so we can't share the answers, but in the quizzes, for the lecture quizzes where you have 2 attempts, having that second attempt show you at least what questions you got wrong, not necessarily the answers, but then you can go and find the answers if you know which ones you got wrong. So, so I'm setting that up so that after your second attempt, you can see what questions you got wrong. So
that's awesome. That's good.
Answers.
Okay.
Yeah, and that way you can see, you'll remember those. And so yeah, we'll do that. And then for anything, if you're not sure why you got the grade you did, yeah, just give me a holler and I can go in and look and see what it's showing in there.
I certainly will. Okay.
All right. So the rest of the measurement unit is just remembering which instruments what. So which one's the beaker, the dropper pipette, the volumetric. So the dropper pipette is just, basically like an eyedropper thing. There's no measure on it. Volumetric has a measure. The pipette pump, you pull water up. Micropipette, could use those tiny volumes and be very precise as we roll them. It's kind of a mechanical thing that we use to draw the liquid in. The Erlenmeyer flask, a lot of people got this wrong on the quiz. That's the one that that kind of narrows toward the top and then has a small tube on the top. So it looks like a graduated cylinder on the top and then flares out at the bottom.
Mm-hmm.
Let's see. I think all of those are straightforward. The ecology lab. Let's see. Oh, here we go.
Ecology was my
lab. That's a good one.
Mm-hmm.
So that one, it's really knowing the terminology. So really going over and remembering the order.
Mm-hmm.
Okay, there we go. Now we're moving again. All right, so lab 4 was pH and solutions. I think the biggest confusion, the biggest problem I see on this is remembering that the scale low numbers are acids. So you can be highly acidic, strongly acidic. That doesn't mean it's a high number. That means it's a low number. The lower you go, the more acidic it is. The higher numbers are the basic numbers. And I think because people think of acidity when they think of the pH scale, they expect it to be the reverse, but the actual measure is a fraction. So how much hydrogen is on the bottom is the denominator of a fraction. So the more hydrogen you have, the smaller the number gets. So that's why pH, that's why the low numbers are acids. And then the definitions of a solution. So a solution is a liquid containing a solute and a solvent. And that becomes really important later when we get to the tonicity, to the osmosis, remembering which one's the solute. The solvent is the water, but how much solute is present, how much stuff is dissolved in the water is the one that is going to determine which way the water flows in osmosis. So remembering solution is a solute and a solvent, that's going to be important both for the pH lab and for osmosis. And knowing that a buffer, this is something else that people, that was commonly missed on the quiz, is that a buffer prevents a change in pH. Or I think it was in Top Hat where I saw a lot of people. So it doesn't try to bring a pH to neutral. It doesn't, because not everything wants to have a pH of 7. So for example, a buffer in our blood would try to keep our blood pH slightly basic, just a little bit basic. It wouldn't want to be neutral because our blood, everything that goes on, those chemical reactions take place, they behave better in a slightly basic solution. So a buffer keeps the pH where it should be. So our stomach should have a pH of about 2. Right? Our stomach is very acidic, so we wouldn't want it to be neutral. We want to keep it at 2. We don't want to get it to get more acidic, but we don't want it to be less acidic. So a buffer would keep it at 2. So it doesn't make it neutral. It keeps it where it should be. It prevents the pH from changing. Any questions on pH?
No.
Okay. The chemical composition of cells. I think this is a pretty fun lab. Testing for all the different things. So on the lab practical, so what the lab practical will be, because normally what we would do is there would be stations set up around the lab, and you would rotate through the stations, and you would see something, some results that you had in lab, or some instrument that you used, or a microscope. There would be something, something there that you had already done or seen. Well, this practical is going to be just like your exams. It's going to be online in D2L, but there's going to be lots and lots and lots of pictures. And the pictures will be of things that you've seen and done on your virtual labs. So knowing what the reaction, what the reagents are and what the reaction is for them, what it looks like. So knowing that Benedict's reagent tests for sugars, for my monosaccharides and disaccharides only. So Benedict's won't give me a positive reaction on a starch because that's a polysaccharide, and this reacts with simple sugars. Sometimes we can get a slightly positive reaction with Benedict's because my polymer, my polysaccharide, is made up of stacks of simple sugars, monosaccharides. So if that starts to break down, like if I heat a solution and I break the hydrogen bonds holding these, the polysaccharides together, then there will be free sugars floating in the solution. So sometimes I can get a slightly positive result, but typically Benedict's is only for testing sugars. And iodine, even though it still tests the carbohydrate, it only reacts to starch. So it's not even a test for all polysaccharides. It's a test specifically for one polysaccharide, starch. So knowing the colors for a positive reaction, the Biarritz is going to respond to proteins or polypeptides. So I have to have more than just single amino acids. They have to be strung together. Then I'll get a positive result. And proteins will give me that strong purple result. You did the emulsification test to break the lipids into smaller little blobs of lipids. So what that looks like. And then the paper test for lipids. Typically in the lab, the paper test is the one that we do every time we go through the drive-up at McDonald's and get French fries. The napkin stays greasy. Right? So that's our lab. Oh, you know what? I jumped. Oh, there we go. Wait, I jumped ahead. Where'd the microscope go? There's the microscope. All right, I thought I'd missed that. So any questions on those tests for the macromolecules or their building blocks? No. So knowing what the building blocks are, what's the monomer, what's the polymer for those that you tested in lab, and what the tests were and what the results looked like. That's going to be the key point there. Let's see. So the microscope. Everybody did well identifying the parts, I think, on the quiz. So it's just remembering what parts, ocular lens, what you look through, objective lens, what's pointed at the object you're interested in. The stage and the mechanical stage. This is easy to confuse, especially if you're not physically manipulating the microscope. The stage is just the platform that I put my slide on. The mechanical stage has the clip that holds the slide, and it's a whole mechanism that moves back and forth and left and right and forward and back to help me position my slide. So there's two separate parts. The stage is just the platform, but the part that actually moves the slide to where I want it to be and helps me find things on it, that's the mechanical stage. Total magnification. This one, we're pretty good on that, but a few people wanted to add instead of multiply. We're going to multiply my ocular lens times my objective lens to get the total magnification. Let's see. As my objective lens numbers go up, my magnification goes up. But as my magnification goes up, my field of view gets smaller. And a good way to kind of remember that is if you're looking at Google Maps, right? As you get closer, as you magnify and zoom in, the area that you see gets smaller. So if you start out looking at all of Charleston, as you increase your magnification, you zoom into your house. So that's the same thing. As you start with a big field of view at low magnification, as you make everything larger and larger, your field of view gets smaller and smaller. Let's see, and then the cells. Looking at the cells, everybody, that's a pretty fun lab. Even the virtual lab is pretty fun seeing everything on there. Questions about the microscope?
No.
All right. Prokaryotes and eukaryotes and cell structures and functions. I think identifying, people did really well. We didn't spend a lot of time on prokaryotes, but just knowing pro means early or before, like the prologue in a book comes before the book. So these are our earlier cells, so they're simpler. So they don't have all those membrane-bound organelles. when you're looking at the models. I think most people did well looking at the models and identifying where things are located. Probably the two that people mix up the most are the smooth endoplasmic reticulum and the Golgi apparatus. Because the rough ER is the one that has bumps all over it from the ribosomes. So everybody's pretty good at recognizing that. But the smooth endoplasmic reticulum and the Golgi apparatus are both membrane-bound sacs that don't have ribosomes on them. So they're easy to confuse. Normally, though, because of the way these work, because how that endomembrane system works, within a cell, because the rough ER is an extension of the nuclear envelope, or closest, the endoplasmic reticulum that's closest to the nucleus is the rough ER. And then out from that, we find the smooth ER. And then the portion that doesn't have ribosomes but is smooth and closest to the plasma membrane. So furthest away from the nucleus is the Golgi apparatus, because its job is to package things to export out of the cell. So it's close to that membrane to get things out of the cell. Let's see, questions on any of the organelles?
What about the chromosomes? I don't see that on here. Is that something? It was in the chapter objectives, but it's not on here.
Yes. So I think that's probably not on here because we're not going to be able to see them under the microscope, but they're in the nucleus, our chromosomes,
Okay.
And we're going to zoom in and spend a lot of time on them in the chapters that are coming up when we get to talking about genetics and inheritance and cell replication, cell division. So we're going to spend a lot of time on chromosomes. So now our chromosomes, they carry our genes. They're made of nucleic acids wrapped around proteins, all that chromatin. They're housed in the nucleus. In the prokaryotes have one single circular chromosome, and they may have a little extra chromosomal bit of DNA called a plasmid. So they'll have one big circle of DNA that has all the information that they need to survive, and the plasmids are just little like bonus DNA, and bacteria will share them. They'll be like, hey, look, this is good. I'm going to make a copy and share it with another bacteria, with one of my friends. Eukaryotes don't have that. We only share DNA through reproduction and make a copy, divide between two cells, or make eggs and sperm, and have that come together at fertilization. So we don't have plasmids. We have chromosomes in the nucleus. Ours are thread-like. And so how many chromosomes we have is one of the ways we identify what species,
male versus female.
we are.
Oh, okay.
So there's 46. So there's 46 chromosomes in humans. So that's how we identify. Oh, we're a human with these 46 chromosomes. There are a couple other organisms, a couple other species that have 46 chromosomes, but they would be like different lengths than ours. They wouldn't look exactly like ours, so we could tell. Oh, those 46 come from some other organism. But the number of chromosomes is distinct for different species. Let's see, differentiating between plants and animals. Did that help? We're going to spend a lot of time on chromosomes.
Oh, yeah, that really did.
Eukaryotes. cellular respiration, all eukaryotes have mitochondria because mitochondria is how organisms get their energy, their ATP. The chloroplast for plants is how they get their food. So animals go out and eat their food. Plants make their food. But then for both plants and animals, they have to do something. They have to get the energy from that food. So the mitochondria is found in both of them. And that's what's coming up on photosynthesis and cell respiration. We'll get into all gory details of that. But plants do have both. So good at identifying plants versus animals.
Yes.
I have a little spinning blue thing on my screen right now. I'm trying to scroll down. Almost to the end. So then the next lab was osmosis. It's might be a painful review to watch for so many of you, isn't it? Because I think it keeps seizing up and you're not going to have very good video. So diffusion and osmosis. Diffusion is the movement of any molecule from high concentration to low concentration. All right, I'll keep going through diffusion.
Okay, I just got back in. I don't know what's going on. I keep having to hang up and come back in.
It's on my end here. So what I did was I instead of setting this, I set this up on my Trident Tech computer. So this actually like connects my home computer to that computer so that I have my files from there. And for some reason, I don't know if they're working on things there, but it's really, really slow and keeps interfering and seizing up. So, So it's on my end. I apologize. I'm sorry. We are almost through here. So diffusion and osmosis.
Oh, I'm listening.
Okay, yep. So diffusion and osmosis. Diffusion, just know it can be any molecule. It can be water, it can be oxygen, it can be carbon dioxide, it can be perfume, it can be anything, any molecule moving from high concentration to low. It can be across the membrane, like solute to 60% solute. With a higher salt, water moves to the hypertonic solution. And then other place to use your words. When we're looking at plant and animal cells. Okay, so animal cells, when they shrivel, it's called crenation. When an animal cell bursts, it lyses, or cytolysis. If it's a red blood cell, we can call that hemolysis. Cyto, that prefix just means cell. Cytolysis, the cell will burst. When I'm talking about plants, I'm not going to use burst or shrink. My cell is not going to burst or shrink in a plant cell because the cell wall is a rigid structure. My cell wall defines my cell in plant cells. What's going to happen is if I put my plant in hypotonic solution, water is going to move into the cell. It's going to fill that central vacuole. It's going to push all the organelles and the plasma membrane up tight against the cell wall, but the cell wall is rigid. So the cell wall is not going to expand. It's going to fill up until the pressure against that wall won't allow any more water to come in. It will become very turgid. Right? It will be filled up. It will be very stiff because it's all full of water. We call that turgor pressure or turgidity. The plant becomes very rigid and firm because it fills with as much water as it can, but then it stops because the pressure of the water on the cell wall is as high as it can get. The cell wall won't let any more water in. Plasmolysis is the opposite. If I put my plant into hypertonic solution and it loses water, the cell's going to pretty much retain its same shape because the cell wall is going to stay the cell wall in that same rectangular shape that it has. But as that water leaves the plasma membrane, it's going to pull away from the cell wall. So what's inside that cell wall, where all the organelles, all of the rest of the cell is, that's going to shrink, but the cell itself is going to stay about the same size. So plasmolysis is the plasma membrane pulling away from the cell wall. So what's inside the cell is shriveling. That central vacuole loses water and shrivels, but the cell's going to stay the same size. So when I'm talking about plants, I want to make sure that I'm using my terms turgor pressure and plasmolysis rather than shrinking or swelling or bursting, because that won't happen in a plant. All right, and then our last lab that will be covered on the exam is enzyme function. So far the quizzes look good. Everybody's doing well on this. On enzymes, again, knowing chemical reaction, how to write a chemical reaction. If my arrow goes left to right, what's on the left side are my reactants. What's on the right side are my products. So what the arrow points to, that's what the chemical reaction produces. Those are the products. So remembering that, reaction on the substrate comes together, the more enzyme I have, the faster those molecules can bump into each other in the reaction occur. Similarly, if I have more substrate, those enzyme and substrate molecules are going to bump into each other faster, and the rate of reaction is going to increase. So as I increase concentration of either one, I'm going to increase the rate of reaction only to a certain point. Is at some point, if I keep increasing the substrate concentration, but not the enzyme concentration, the enzymes are going to be working as fast as they can. They're going to grab on every single one of those enzymes is going to be busy converting substrate. So I'll reach a plateau. If I optimal pH. So it's not always 7. Typically it's about 7 for some. As mentioned, our blood pH is slightly basic. The enzymes that work in our bloodstream work at a slightly basic pH. As I get more and more acidic, the reaction will slow down. If I get too acidic, those hydrogen ions are going to interfere with the hydrogen bonds holding the enzyme's shape together, and it will denature, and the reaction will stop. Similarly, if I get too basic, if my pH increases too much, same thing will happen. So the effect of pH on enzyme activity is dependent. It's all around a point. So if this is my optimal pH, as I get higher or lower, my rate of reaction falls off. So I get a bell-shaped curve for how fast that reaction is, and at some point, we denature the enzyme if we get too far away from the optimal pH, and the reaction stops. The experiment you did in lab, remember the enzyme is catalase. Easy to get that term confused with, oh, all enzymes are catalysts, but not all enzymes are catalase. That ending ASE usually indicates an enzyme. Most enzymes, but not all, end in ACE. Catalase is the enzyme that breaks down hydrogen peroxide into water and oxygen. And that oxygen in the water is what causes the bubbles that we see. So catalase, my substrate, what it acts on is hydrogen peroxide. What it produces is water and bubbles. So that's my product. And look at the equation. What the arrow is pointing at, those are my products. That's what's produced. And what I start with, that's my substrate or my reagent. And enzymes can work both ways. They can take small molecules and